In 2003, scientists first sequenced the human genome. This came 50 years after the structure of DNA was described. Today, we're going to learn about both of these discoveries in science. Inside our cells, the nucleus contains chromosomes. In the meiosis video, we learned that our body cells have 46 chromosomes. Every chromosome is made up of DNA, and DNA is made up of lots of small units, so we can call it a polymer. It is made from two strands that form a double helix. This is a definition you need to learn. Now let's look at genes. A gene is a small section of DNA that codes for a specific sequence of amino acids, and these make a specific protein, for example, the pigment that colors your eyes. You'll learn a lot more about how genes work in the next video. As I mentioned at the start, scientists have sequenced the human genome. But what is a genome? Well, it is simply the entire genetic material of an organism. This includes all the DNA inside the nucleus and the DNA found inside the mitochondria. Understanding the human genome is an important area of research. Scientists are using it in 3 ways. Number 1, they're using it to improve their understanding of inherited disorders and how to treat them better. This includes looking at cystic fibrosis and sickle cell anemia. Number 2, they're using it to search for genes linked to disease. This includes diseases like heart disease and type 2 diabetes. To do this, they're looking for patterns in the genomes and trying to spot any similarities. And finally, number 3, they're using genomes to trace past human migration patterns to look at how we moved across the world. Okay, let's try some questions. Pause the video, give them a go, and press play when you're ready to go over them. 1. Describe the structure of DNA. It is a polymer made of 2 strands that form a double helix. 2. Complete. A gene is a small section of DNA. Each gene codes for a specific sequence of amino acids, which make up a specific protein. 3. What is the genome of an organism? It's the entire genetic material of that organism. And 4. Suggest how sequencing human genomes could allow scientists to identify genes that increase the risk of diseases. Okay, so by comparing genomes of sufferers to genomes of healthy people, scientists can spot which genes are different and can test these to see if they are responsible for the increased risk of disease. Take a look and notice I've used the same language in my answer as is given in the question, talking about the risk of disease, not just saying disease. Alright, how did you do? Understanding how genes interact is really interesting, and it can explain how 2 black fur rats can have a brown fur rat baby. We're going to learn about this in the next video of inheritance, and please subscribe if you're finding this useful. Thanks and bye!